Hello, my name is William Grant Hale Jr. Today I'm going to talk about the different isoforms of aldolase and their different expression patterns. Okay, in the introduction for this, aldolase will be described as to what it is, its history, specificity, composition, and molecular characteristics. I will be talking about the three different types of aldolase that will be covered: aldolase A, which is aldo A, aldo B, and aldo C. And finally, I'll be talking about the aldolase gene expression for the Xenopus laevis will be covered as well, and that's actually an African clawed frog. So hopefully you will stay tuned. So now we're talking about what is aldolase. Aldolase is present in all animals and plant tissue and in most microorganisms. Class 1 aldolase, found in animal and higher plant tissues, is characterized by the lack of requirement for a bivalent metal cofactor and by the formation of ketamine shift base. The class 2 aldolase are found in the lower animals, the prokaryotes and the lower eukaryotes, and it requires the bivalent metal cofactor. There are three types of class 1 aldolases. Type A, the major form, is found in the muscle. Type B is in the liver and kidneys, and type C and A is found in the brain as well. Now we'll talk a little bit about the history of this as well. Aldolase history in brief. In 1935, aldolase was found to be present in muscle and yeast. Muscle aldolase was first separated from isomerase in 1940. First, it proposed the presence of the aldolase in bacteria in 1934, which was later confirmed. And in the 1950s and 1960s, the reaction mechanism was investigated, and the classification scheme that divided aldolase into class 1s and class 2s was developed. Then work was begun to be investigating the inhibitors began in the mid-1960s and early 1970s. But in 1969, the crystals of aldolase were studied in order to determine the subunit structure. The first low-resolution crystal structure was determined in 1985 and was refined in 1987. Then it talks about, after some debate over mechanism and the involvement of the particular active site residues, it was determined that in 1993 that a cysteine residue was not present at or near the substrate binding site, despite initial studies concluding otherwise. The first high-resolution structure of a rabbit aldolase was reported in 1997. Today, work investigating aldolase inhibitors continues. Studies are also being conducted to better understand its utility in chemoenzymatic synthesis, its interactions with other cellular proteins, and its reaction intermediates. Its specificity. Aldolase is highly specific for its substrate D, fructose. As we said before, there's actually three isomers or isoforms of aldolase. The three isozymes A, B, and C found in mammals are composed of four identical subunits, each a polypeptide chain of 360 amino acids. The enzyme has a homoheteromeric structure with subunits that are approximately 40,000 Daltons in size. The structure of these subunits is a beta barrel surrounded by alpha helices and connecting loops. If activity of one of the four units of the homoheteromeric is knocked out, the entire complex is incapable of normal function, and indicating a tight communication between these active sites. In the molecular composition, the primary structures of class 1 eukaryotic enzymes are highly conserved. However, there are no similarities between the sequences of class 1 and class 2 enzymes, as they have separate evolutionary origins. However, both class 1 and class 2 enzymes contain a carboxy terminal tyrosine residues that are required for maximal catalytic activity. Prolonged contractile activity reduces the type of aldolase expression in messenger RNA by downregulating cytoplasmic glycolytic enzymes and enhancing mitochondrial enzymatic activity. Now we're going to actually go into the three different types. First, what we're covering is aldolase A. also known as aldo-A. It's also known as fructose bisphosphate aldolase, and it's an enzyme that in humans is encoded by the aldo-A gene on chromosome 16. The protein encoded by this gene is a glycolytic enzyme that catalyzes the reversible conversion of fructose 1,6-bisphosphate to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These three aldolase isosomes A, B, and C, encoded by 3 different genes, are differentially expressed during development. Aldolase A is found in developing embryos and is produced in even greater amounts in adult muscles. Aldolase A expression is expressed in the adult liver, kidney, and intestines, and similar aldolase C levels in the brain and other nervous tissues. Aldolase A deficiency has been associated with myopathy and hemolytic anemia. Alternative splicing, alternative promoter usage results in multiple transcript variants. Related pseudogenes have been identified on chromosomes 3 and 10. Aldolase aldo-A is one of the glycolytic enzymes primarily found in developing embryo and adult muscle. Recently, a new role of aldo-A in several cancers has been proposed. However, the underlying mechanism remains obscure and inconsistent. It was investigated that aldo-A associated with its genes, the AA genes, using available microarray data sets to help elucidating the role of aldo-A in cancers. Aldo-A could contribute to the progress of cancer at least partially through its association with genes relevant to cell cycle independent of glycolysis. These genes plus aldo-A represent a potential new signature for development and prognosis in several cancers. Metabolic dysfunction is a hallmark of cardiac hypertrophy and heart failure. During cardiac failure, the metabolism of cardiomyocytes switches from fatty acid oxidation to glycolysis. However, the roles of these key metabolic enzymes in cardiac hypertrophy are not fully understood. In the present work, it was identified that aldo-A, as a core regulator of the cardiohypertrophy, the messenger RNA and protein levels of aldo-A were significantly upgraded in transverse aortic constriction and isoproteirenol induced hypertrophic mouse heart. Overexpression of aldo-A in cardiomyocytes promoted ISO-induced cardiomyocyte hypertrophy, whereas aldo-A knocked down repressed cardiomyocyte hypertrophy. In addition, the adeno-associated virus 9 mediated in vivo knockdown of aldo-A in the heart, rescued ISO-induced decrease in cardiac ejection fraction and fractional shortening in repressed cardiac hypertrophy. Mechanism study revealed that aldo-A repressed the activation of the dependent protein kinase, signaling in a liver kinase, B1, dependent and independent manner. Inactivation is a core mechanism underlying aldo-A mediated promotion of the ISO-induced cardiomyocyte hypertrophy. By contrast, activation of this metformin, blocked the aldo-A function during cardiomyocyte hypertrophy, and it continues on with that. Now we're going to go on to aldolase B. Aldolase B. Aldolase B is also known as fructose bisphosphate aldolase B or liver type aldolase in one of the 3 isoenzymes A, B, and C of the class 1 fructose 1,6-bisphosphate aldolase enzyme, and plays a key role in both glycolysis and gluconeogenesis. The generic fructose 1,6-bisphosphate aldolase enzyme catalyzes the reversible cleavage of fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, as well as the reversible cleavage of fructose 1-phosphate into glyceraldehyde and dihydroxyacetone phosphate. In mammals, aldolase B is preferentially expressed in the liver, while aldolase A is expressed in muscle and erythrocytes, and aldolase C is expressed in the brain. Slight differences in isozyme structure results in different activities for the two substrate molecules, FBP and fructose 1-phosphate. Aldolase B exhibits no preference and thus catalyzes both reactions, while aldolase A and C prefers FBP. In humans, aldolase B is encoded by the ALDO-B gene located on chromosome 9. The gene is 14,500 base pairs long and contains 9 exons. Defects in this gene have been identified as a cause of hereditary fructose intolerance. Continuing on with ALDO-B, the rise of fructose consumption and its correlation with symptoms of metabolic syndrome has highlighted the need for a better understanding of fructose metabolism. To that end, valid rodent models reflecting the same metabolism as in humans, both biochemically and physiologically, are critical. A key to understanding any type of metabolism comes from study of disease states that affect such as metabolism. A serious defect of fructose metabolism is the autosomal recessive condition called hereditary fructose intolerance, we've talked about before, caused by mutations in the human aldolase B gene. Those afflicted with this experience liver and kidney dysfunction after fructose consumption, which can lead to death, particularly during infancy. With very little or low levels of fructose exposure, patients develop non-alcoholic fatty liver and fibrosis. Sharing liver pathology is also seen in MBS. A lot of times you have fatty livers if you're an alcoholic. Continuing on with aldolase B. Deregulation of aldolase B has been reported in hepatocellular carcinoma. However, its clinical significance, its role in pathogenesis, this remains largely unknown. The downregulation of ALDO-B could indicate a poor prognosis for HCC patients, and therefore ALDO-B might be considered a prognostic biomarker for HCC, especially at the early stage. In addition, ALDO-B inhibits the invasive features of cell lines partly through TET1 expression. Now we'll go to aldolase C. Aldolase C, the third type, is also known as fructose bisphosphate aldose, is an enzyme that in humans is encoded by the aldose gene on chromosome 17. This gene encodes a member of the class 1 fructose bisphosphate aldolase gene family, expressed specifically in the hippocampus and the Purkinje cells of the brain. The encoded protein is a glycolytic enzyme that catalyzes the reversible aldo cleavage of the fructose 1,6-bisphosphate and fructose 1-phosphate to dihydroxyacetone phosphates in either glyceraldehyde 3-phosphate or glyceraldehyde respectively. The amino acid sequence of aldose C is highly similar to those in the other isosomes, sharing a 68% identity with aldo-B and 78% identity with aldo-A. They are conserved in the active sites on the 3 isozymes. This active site is located in the center of the homoheteromeric alpha-beta barrel structure of these aldolases. However, several structural details set aldose apart. For instance, some adopt an intermediate conformation between the liganded and unliganded structures observed in the other isosomes. Also, the C-terminal region between these forms a salt bridge with the barrel region, that is absent in both the A and the B isoforms. Moreover, the electrostatic surface of aldose C is more negatively charged, which may serve as an acidic binding site or as a docking site to accommodate the C-terminal conformations. Continuing on with aldose. Aldose has long been used as a marker for the study of cerebellar compartmentalization due to its specific expression in distinct subpopulations of cerebellar Purkinje cells that are arranged in functionally significant longitudinal stripe-shaped compartments. Each longitudinal compartment, determined by the striped aldose C expression pattern, is innervated by climbing fibers originally from a specific subarea of the inferior